Dear students, in this lecture we'll learn about the analysis of restriction enzyme site available in the gene sequence you are going to clone. So there are various online tools available for this purpose like NEB cutter, web cutter, mapper, web map, restriction mapper. NEB cutter is user friendly, we are going to use that. So for that you have to type NEB cutter. Click on NEB cutter V20. You need to either paste the sequence which is available with you in FASTA format, or you can paste the gene bank number, accession number, or you can directly paste the sequence after getting it from the NCBI. So I'll use the third option. I'll go to NCBI, click on nucleotide. I'll type my favorite sequence name. Click on complete CDS. You can take accession number from here also, and or else you can get the FASTA format of the nucleotide sequence. Copy this sequence, paste it into NEB cutter. Now you have different options. You have pull down menu of different vectors available with this. So I'll leave it as such. Viral phase and NEB cutter, NEB enzymes, all commercially available specificities, all specificities, all defined oligonucleotide sequences, only defined oligonucleotide sequences. You can choose the option according to your requirement. Here you can check if the sequence you are putting it linear or circular. Our sequence is human gene, so it is linear, so I'll leave it as such. And minimum ORF length to display. So this is for amino acid sequence. So if your gene sequence, amino acid sequence is more than 100 amino acid or less than that, you can change the number or leave it as such. You can give the name to the sequence, and you can disable NEB cutter cookies and delete projects. And from here you can set the colors. So these are the color scale available. For blunt you can choose red, for 5' extension you can use royal blue, 3' extension you can use green, or you can change the color from here. Or there are more options available. We'll check what options are there. So you have type 1 and type 3 enzymes, homing endonucleases, nicking enzymes, and there are a few more options. Ignore CpG methylation, ignore EcoB1 methylation, ignore DAM methylation, ignore EcoK1 methylation, ignore DCM methylation. So these are all options given to you which you can choose according to your requirement. We'll go one by one. Type 1 and type 3 enzymes. So you know that type 2 enzymes are commonly used for cloning purposes, and they have restriction site in which they cleave itself in the restriction site. While type 1 enzymes have a specificity that they cleave at the remote location from the recognition site, and they also additionally have methylase activity as well as ATP dependent. They are ATP dependent. And another option, like another enzyme is type 3 enzyme. They have same site, like they will cleave in the same restriction enzyme, but in addition they have methylase activity and they are also ATP dependent. So these enzymes, if you click this, this will also give you type 1, type 2 and type 3 enzymes list. All enzymes will be given to you. Then another option is homing endonuclease. The endonuclease which cleave the genomic DNA within the cell, synthesizing them. So if you click this checkbox, this will give you the homing endonucleases as well. Then there are nicking enzymes. So these enzymes which cut only one strand of double-stranded DNA at a specific recognition site. So if you click this, this will give you a list of enzymes which are having these specificity. Apart from that, if you want to ignore some input sequences, then you can click on this, like input, ignore CpG methylation, ignore EcoB1 methylation, ignore DAM methylation, ignore EcoK1 methylation, ignore DCM methylation. So what it offers, so if you click on this, They will ignore while screening for overlapping methylation sensitivity of the enzyme. Methylation sensitive restriction enzymes cannot cleave methylated cytosine and thus are used to analyze methylated DNA and the methylation status of cytosine residue in the CpG sequences. Similarly, these are other options available. Now there is another option, sequence is a fragment. So if you click this, this box you have to click when your sequence you are going to clone is a fragment of a particular gene or particular full length sequence. Then you can click this, or you can give the option by writing the number of the bases from here to from there, so that you can select that particular portion in which your sequence you want to check for the availability of restriction enzymes. So I'll just click on submit button here. So you see there are number of options available, and apart from that your sequence is cleaved and it is showing different restriction sites available in the protein or a gene sequence, you can see here. So there are a few options like new DNA. If you click this, you will be directed to the initial page, and if you click on custom digest, so if you click on this will give you opportunity to set a restriction enzyme, so that if you want to check a particular restriction enzyme, then you can click on this will tell you whether the sequence is available in the sequence of your interest or not. Restriction enzyme sequence is available in the sequence of your interest or not. Next option is view sequence. So you can get this ORF summary, which gives you different options like coordinates of the gene, length of polypeptide, and gene bank protein ID of the respective gene sequence, single cutter restriction enzymes, etc. And then you have option of saving your project, and then you can print it. Now there are other option, availability, all commercial, all. So if you click on all commercial, this will display the restriction enzyme which are commercially available from any agency. So although this is default set for NEB produced restriction enzymes, and all, if you click on this will display all commercial appropriate literature cited but not commercially available restriction enzymes. Next option is display. So 1, 2 or 3 cutters. So if you click on these cutters, this will tell you, like if you click on 2 cutters, it will give you the list of enzymes which are cutting at 2 sides in the fragment, and it will cleave the fragment into 3 parts. Then you have 3 cutters, so this will give you a list of enzymes which are having 3 cutting sides, and it will cleave the sequence into 4 parts. So you are interested in more 0 cutters, so you will click on this. Alright, so this will give you a list of restriction enzymes which are not having any site available in the enzyme in the protein sequence or in the gene sequence, so that you can choose the enzyme from this list based on the availability of the restriction enzyme in your vector of interest. Also you have to see whether the enzyme is available easily and it is easy to handle, it is easy cutter in general uh, and in your laboratory. What are other option? We'll just see. There is zoom in option. This enlarges the selected region. So you have to like select a region here, zoom in, so this way you can click and zoom. So unzoom, I'll just unzoom. So this is going back to the original site. Now there is other option, all sites. So it will enlist all the restriction enzyme sites according to their location along the input sequence. All type of enzymes will be given there. Then save all sites. So this will save the sites in your computer, in your system, in the text format. So there is another side option, flanking enzyme. This is useful for genetic studies. So in this you can identify the restriction enzyme for the regions flanking a target region on the input sequence. So we'll see your assignment for today, that you can check the gene of your interest in this NEB cutter and see what restriction enzyme you can use based on the vector you are going to utilize in your laboratory.